Welcome back to this lesson on gene regulation and the Lac Operon. Learning goals to be achieved are 1. Recognize the importance of gene regulation. 2. Distinguish positive regulation from negative regulation. 3. Identify similarities and differences in gene regulation in prokaryotes and eukaryotes. And 4. Understand the Lac Operon in E. coli as a gene regulation model in prokaryotes. With a couple of exceptions, all the cells in our body contain the exact same DNA. And despite this, we know our cells look so different and perform many different functions. So how can that be possible? Well, the answer is gene regulation. Regardless of whether it is in a simple unicellular prokaryotes or a complex multicellular eukaryotes, each cell controls when and how its genes are to be expressed or transcribed into an mRNA and then ultimately translated into a protein. The process of controlling which genes in a cell's DNA are expressed is called gene regulation. Or in other words, it is the process of turning genes on or off. Gene regulation ensures that appropriate genes are only expressed when needed. A significant amount of energy is required for a gene to express, and so it would save energy if genes are expressed only when they are required. Gene regulation also allows cells to adjust to changing environmental conditions. There are several examples of organisms changing their gene expression in response to environmental changes. To give you one such example is the difference in the expression of the fur color gene in the Himalayan rabbit at different temperature. In the central parts of the rabbit's body, where the temperature is warmer, around more than 35 degrees Celsius, the fur color gene is inactive and no pigments are produced, causing the fur color to be white. But in the regions like the ears, tip of nose, and feet, where the temperature is relatively lower, the gene actively produces pigments and thereby causes these parts to become darker or black. Above all, gene regulation in multicellular organisms facilitates differentiation and division of labor among cells. Let's take an example. These are RBCs which appear red due to the presence of hemoglobin. And over here on the right is a brain cell. Now they look highly different and they have different functions. But we are aware of the fact that they have the exact same DNA. Practically, every single cell in our body contains the genes for hemoglobin. So why is it that only RBCs are able to actually synthesize hemoglobin? It's all due to the process of differential gene expression. Hemoglobin gene is activated in RBCs and not in brain cells or muscle cells or any other cells for that matter. Likewise, the gene for dopamine, which is one of the happy brain chemicals, is expressed in the brain cell and not in the RBCs or other cells. So an important point to realize is that the difference between cells is not due to the genes, but rather it is the differential expression of the genes. The cells in multicellular organism can look different and perform specific functions by expressing very different sets of genes. So how is gene expression regulated? Cells use a variety of mechanisms to increase or decrease the expression of a specific gene. And the complicated task of gene regulation is accomplished primarily by the different regulatory proteins. These regulatory proteins can either increase transcription or they can suppress transcription by binding to a specific region in the DNA. Regulatory proteins can subject a gene to a positive regulation or a negative regulation. In positive regulation, the regulatory protein is an activator which binds to a specific DNA sequence and promotes transcription. Activator, therefore, is a regulatory protein that turns a gene on. In contrast, when a gene is under negative regulation, the regulatory protein is a repressor which blocks or suppresses transcription. Repressor therefore turns a gene off. That said, some genes are always active or on without any regulation, and these genes are called as constitutive genes. The genes for ribosomes are an example for this. Ribosomal genes are continuously transcribed because ribosomes are constantly needed for protein synthesis. Another example are the genes called as housekeeping genes. These genes are continuously expressed in all cells of an organism since they are essential for the basic cellular functions. And we will also get to know one constitutive gene in prokaryotes in a while. Well, next, speaking of the similarities in gene regulation between prokaryotes and eukaryotes, there are some basic similarities between them. RNA polymerase in both prokaryotes and eukaryotes binds to the promoter sequence in order to initiate transcription or to initiate gene expression. Moreover, both prokaryotic and eukaryotic DNAs have binding sites for the regulatory proteins to control when and how its genes are expressed. At the same time, there are differences in gene regulation between them. It turns out that the default state of gene expression in prokaryotes is on. This is because RNA polymerase in prokaryotes can easily access any promoter in a DNA strand since the DNA is not associated with nucleosomes. In eukaryotes, however, the default state of gene expression is off. So why is this the case? Well, as you know, the DNA in eukaryotes is packaged into nucleosomes, that is, our DNA is tightly wrapped around the histone proteins. So the coiling and supercoiling of DNA around the histone proteins restricts access to the gene. If you can recall, we have learned of the two types of chromatin fibers based on their compaction. Can you remember? We classified them as euchromatin and heterochromatin. So in the loosely packed euchromatin, since the genes are accessible for transcription, such regions are referred to as transcriptionally active regions. The genes in the dense heterochromatic regions are not accessible for transcription and hence they remain inactive. So for gene expression to occur, eukaryotes must modify its dense heterochromatin into a more open beads on string euchromatin form to expose the gene for transcription. A second difference between prokaryotes and eukaryotes is in terms of how genes are organized. In prokaryotes, genes with similar functions are grouped together and organized into an operon. The related genes in an operon are turned on or off together, and such regulation is referred to as co-regulation. Operons are not found in eukaryotes, and in eukaryotes, each gene has its own promoter. Thirdly, the regulation of gene expression in prokaryotic cell occurs at the transcriptional level, while in eukaryotes, gene regulation is more complex. It could be regulated at the transcriptional level, at the RNA processing level, at the translational level, and also at the post-translational level. Of these different stages, gene expression is predominantly regulated at the transcriptional level in eukaryotes. As per our course requirement, we need to understand and apply the concept of gene regulation to a specific gene model, the Lac Operon in E. coli. And first thing first, let me explain what a Lac Operon is. As I said before, an operon refers to a group of genes with a single promoter. A Lac Operon is specifically consisting of a group of 3 genes with a promoter. And the 3 genes encodes for 3 different proteins involved in lactose metabolism. Regulation of the Lac Operon was first studied by Jacob and Monod in 1962. Structurally, The Lac Operon is made up of a regulatory gene, a promoter, an operator, and 3 structural genes. The 3 structural genes of the Lac Operon are referred to as LAC-Z, LAC-Y, and LAC-A genes. The LAC-Z gene encodes for beta-galactosidase, an enzyme primarily responsible for the hydrolysis of lactose into its 2 monosaccharides. The LAC-Y gene encodes for permease, which facilitates the diffusion of lactose into the cell, and the LAC-A gene encodes trans-acetylase enzyme, which is an enzyme whose role in Lac Operon is unclear. Altogether, these 3 gene products acts to break down lactose into its 2 monomeric subunits, glucose and galactose, to be used as an energy source. Expression of the 3 structural genes are under the control of a regulatory gene LAC-I at the 5' end of the operon. LAC-I gene is not under any regulation and is a constitutive gene, which means LAC-I is continuously transcribed into a repressor mRNA, which is ultimately translated into the repressor protein. The LAC repressor protein binds to a specific sequence in the operon called the operator sequence. The operator sequence overlaps with the promoter sequence and is located between the promoter and structural genes. Now, binding of the repressor protein gets in the way of RNA polymerase and inhibits transcription of the LAC genes. The Lac Operon is thus under negative co-regulation, which is to say that the active repressor prevents transcription of LAC-Z, LAC-Y, and LAC-A genes altogether. So how is the Lac Operon turned on then? Well, we know that Lac Operon is required by E. coli to use lactose as an energy source. However, lactose is not their preferred source of energy. If glucose is available to them, they would rather use glucose over lactose. You know, glucose is the simplest monosaccharide. It is therefore easier to release energy from glucose than lactose. Bottom line is, E. coli turns on the Lac Operon only when glucose is not available and lactose is available. Let's now see how lactose affects the Lac Operon. When lactose is absent in the medium, the active repressor protein binds to the operator sequence and inhibits transcription of the 3 LAC genes. Lac Operon therefore remains switched off. When lactose is present in the medium, its metabolite called allolactose binds to the repressor protein and inactivates the repressor. The inactive repressor is not capable of binding to the operator anymore and clears the way for RNA polymerase to smoothly transcribe the 3 LAC genes into 1 polycistronic LAC mRNA. Allolactose or lactose is called an inducer of the Lac Operon because it induces or turns on the expression of the LAC genes. The transcribed polycistronic LAC mRNA is finally translated into the 3 different enzymes or the gene products, which are beta-galactosidase, permease, and trans-acetylase, which collectively functions to break down lactose and release energy. You might think that the repressor protein and lactose are the only 2 factors that interact to regulate the expression of LAC genes in E. coli. Well, I must clarify that it doesn't end here. There are other molecules that play equally significant role in the regulation of Lac Operon, such as glucose. In the presence of glucose, Lac Operon is turned off. And this makes sense because there's no need to metabolize lactose when glucose is readily available in the medium. So you can clearly see how smart and energy efficient the cells are in their functioning. On the other hand, When glucose is absent and lactose is present, the Lac Operon is turned on and there is high level of transcription of the Lac genes to utilize lactose. So to sum up, we can clearly make sense of the importance of gene regulation. Through the process of turning the expression of genes on and off by regulatory proteins, wasteful expression of genes are avoided and proper differentiation and division of labor are facilitated among cells. Any malfunction or unregulated gene expression can prove detrimental to the cell and can lead to the development of many diseases, including cancers.